Hi, my name is Carlyn Cefaratti, and I was given Group 1 for my presentation. So I chose Topic 1, Membrane Physiology, to present on. And my name is Carlyn Cefaratti. In the bottom right of this image, you see a cell membrane. Just because that's mostly what we're going to be talking about in this presentation, I want to get a visual before I get into it. The question was to describe how the structure, lipids, and proteins of a cell membrane facilitates its function, i.e. being semi-permeable. The cell membrane, also known as the plasma membrane, is a vital component of all cells. Its structure is crucial for its function as a semi-permeable barrier, allowing certain substances to pass through while restricting the movement of others. The cell membrane mostly consists of lipids and proteins, which work together to facilitate its function. Lipids are the primary component of the cell membrane and include phospholipids. Phospholipids have a hydrophilic head and a hydrophobic tail. These phospholipids arrange themselves in a bilayer with the hydrophilic heads facing outward towards the environment, while the hydrophobic tails face inward, shielding themselves from the water. This arrangement creates a selectively permeable barrier that controls the movement of the substances across the membrane. As for the proteins, they play a crucial role in the structure and the function of the cell membrane. They're filled with lipid bilayers and can be classified into two categories. They're either integral proteins, which are proteins that span the entire lipid bilayer, with portions extending into the extracellular and intracellular regions. They have hydrophobic regions that interact with the hydrophobic tails of the phospholipids, anchoring them in the membrane. Integral proteins serve various functions, including transport of some molecules across the membrane and cell signaling. Peripheral proteins. These proteins are located on either side of the lipid bilayer, associated with the surface of the membrane. They interact with the polar heads of the phospholipids or with integral proteins. Peripheral proteins participate in various cellular processes, such as cell adhesion, cell signaling, and providing structural support to the membrane. The fluid mosaic model includes the arrangement of lipids and proteins in the cell membrane following the fluid mosaic model. This model suggests that the components of the membrane are not rigidly fixed, but rather have a dynamic nature. The phospholipids can move across with the bilayer, giving the membrane proteins to diffuse with the lipid matrix. Selective permeable. The semi-permeable of the cell membrane is primarily due to its lipid bilayer and associated proteins. Small uncharged molecules such as oxygen and carbon dioxide can freely diffuse through the lipid portion of the membrane. However, polar or charged molecules such as ions cannot pass through the hydrophobic lipid bilayer. For the movement of such molecules, specific transport proteins are responsible. These integral proteins form channels or carriers that facilitate the transport of substances across the membrane. I added a picture of a lipid-anchored protein to the bottom of the slide to show what it would be seen as to the naked eye. To define diffusion, I took it to a deeper level. Diffusion is a passive process by which molecules or particles move from an area of higher concentration to an area of lower concentration. It is driven by the random thermal motions of particles and does not require the input of energy. In other words, diffusion occurs due to the tendency of particles to spread out and achieve a more uniform distribution. During diffusion, molecules move down their concentration gradient, which is the gradual change in concentration from one region to another. The concentration gradient represents the difference in the concentration of a substance between 2 points. Diffusion allows substances to move across cell membranes or within a fluid medium, such as air or water, until they are evenly distributed. The rate of diffusion is influenced by several factors, including the concentration gradient, temperature, speed of molar motion, surface area available for diffusion, and the size and nature of the particles involved. Small, non-polar molecules typically diffuse quicker than larger or polar molecules. It is an essential process in various biological systems. It allows for the exchange of gases in the lungs, the transport of nutrients and waste products across cell membranes, and the movement of molecules within cells and throughout the body. Diffusion plays a vital role in maintaining the balance and functioning of biological systems. I added a picture of diffusion being processed over time to the left. Compare and contrast active and passive transport mechanisms across the cell membrane. Active and passive transports are two distinct mechanisms by which substances are transported across the cell membrane. Let's compare and contrast. Passive transport, which means a process by which substances move across the cell membrane without the input of energy. Energy requirements is no energy required as the movement occurs along the concentration gradient from a higher concentration to a lower concentration. Diffusion, facilitated diffusion, and osmosis, selectivity, all are involved in the passive transport across the membrane. Examples of passive transport include the diffusion of oxygen and carbon dioxide across the respiratory membrane, the facilitated diffusion of glucose through glucose transporters, and the osmosis of water across the cell membrane. On the other hand, for active transport, which means a process by which substances are moved across the cell membrane against their concentration gradient, requiring the input of energy, usually in the form of ATP, is required to drive the transport process against the concentration gradient. There's primary active transport, secondary active transport, selectivity, and some examples would be examples of active transport include the sodium-potassium pump, which transports sodium ions out of the cell and potassium ions into the cell, and the sodium-glucose co-transporter in the small intestine, which both use the transport of glucose with the movement of sodium ions. The key difference between active and passive transport include the requirement of energy, the direction of movement relative to concentration gradient, selectivity, and the specific mechanisms involved. Passive transport occurs along the concentration gradient without energy input, while active transport requires energy to move substances across their concentration gradient. And I added a picture of active and passive transport, and it's kind of comparing the two of them at once. Compare and contrast channel-mediated and carrier-mediated transport. Channel-mediated and carrier-mediated transport are two mechanisms by which substances are transported across cell membranes. So then let's compare them. For channel-mediated transport, which means a transport that is a process by which substances move across the cell membrane through protein channels. Protein structure form water-filled pores or channels in the membrane, allowing specific molecules or ions to pass through. Their selectivity is high as they are specific to certain molecules or ions based on their size, charge, and shape. The mode of the transport is a channel-mediated transport facilitated by the passive movement of substances down their concentration gradient, and it is not necessary to have energy of any type. Their transport rate is high as substances can move rapidly through the open channels, and some examples of channel-mediated transport include the movement of ions such as sodium, potassium, calcium, and chloride through ion channels. For carrier-mediated protein, which is a process by which substances are transported across the cell membrane with the help of carrier proteins, their protein structure undergo conformating changes to bind with specific molecules or ions, allowing them to be transported across the membrane. And their selectivity is also high as they have binding sites that are specific to particular molecules or ions. The carrier-mediated protein mode of transport can facilitate both passive and active transport, depending on the concentration gradient and energy requirements. For their passive transport, some carrier proteins can facilitate diffusion, where substances move down their concentration gradient without the input of energy, as we just learned. And then they can also actively transport substances against their concentration gradient, requiring the input of energy in the form of ATP, which we also just went over. The carrier-mediated transport genuinely exhibits a lower transport rate compared to channel-mediated transport, as the conformational changes included in binding and transports can be lower. Examples of carrier-mediated transport include the facilitated diffusion of glucose through glucose transporters and the active transport of ions such as sodium and potassium through ion pumps. The key differences between channel-mediated and carrier-mediated transport include the mechanisms of transport, protein structure, selectivity, mode of transport, transport rate, and specific examples. Channel-mediated transport involves the movement of substances through protein channels, is highly selective, and primarily facilitates passive transport. Although, in contrast, carrier-mediated transport involves the binding and transport of substances by carrier protein, is also highly selective, and can facilitate both passive and active transport. I added an image of the carrier proteins moving across the transport, and then you can obviously see what's going on here. Describe the general differences between leak channels, gated channels, and pumps. So right off the bat, leak channels, gated channels, and pumps are all types of membrane proteins involved in the movement of substances across cell membranes. Between the three types, we can start off with leak channels, which you can find in the image all the way to the left of the screen. Leak channels' function is that they are always open or have a baseline level of permeability, allowing the passive movement of specific ions or molecules across the membrane. For regulation, leak channels are not regulated by any specific signal or stimulus. They maintain a continuous flow of ions or molecules based on their concentration gradient. For their selectivity, leak channels exhibit selectivity based on size and charge, allowing specific ions or molecules to pass through while restricting others. Examples of leak channels include leak potassium channels, K+, leak sodium channels, Na+, and leak chloride channels, Cl-. As we move on to gated channels, which is the image in the middle of this presentation, their function: gated channels are protein channels that can be opened or closed in response to specific signals or stimuli, regulating the movement of ions or molecules across the membrane. For their regulation, gated channels have mechanisms for opening and closing, such as changes in voltage, binding of ligands, or changes in mechanical stress. For their selectivity, gated channels also exhibit selectivity based on size and charge, much like the leak channels, but they also pass through while restricting others. Examples of these channels include voltage-gated sodium channels, ligand-gated ion channels, and mechanosensitive channels. As we move on to pumps, I want to show that a sodium-potassium pump is shown in the image all the way to the right of the screen. The function of pumps are membrane proteins that actively transport ions or molecules across the membrane against their concentration gradient, requiring the input of energy, which is usually in the form of ATP. Their energy requirement is that pumps utilize energy to move substances against a concentration gradient, maintaining concentration gradients and contributing to various cellular processes. Regulation of pumps, which they are regulated by factors such as the availability of energy in the form of ATP, ion concentrations, and other cellular signals. For selectivity, pumps also exhibit selectivity, typically transporting specific ions or molecules across the membrane. Examples of pumps include the sodium-potassium pump, which you see on the screen to the right, which transports sodium ions out of the cell and potassium ions into the cell, and then the calcium pump, which transports calcium ions out of the cell or intracellular compartments. Leak channels are continuously open and allow the passive movement of ions or molecules. Gated channels can be open or closed in response to specific signals or stimuli. Pumps actively transport substances against their concentration gradient, requiring the input of energy. Each of these mechanisms contribute to the regulation and maintenance of ion and molecule movement across cell membranes. Let's move on. The question says, integrate how the structure of a cell membrane results in selectively permeability of molecules. So obviously I added a picture of a cell membrane like I did in the first slide, just so that after learning the information, we're still aware of what it looks like. The selective permeability of molecules across a cell membrane is a result of its specific structure and composition. The structure of the cell membrane, primarily composed of lipids and proteins, contributes to its selective permeability in the following ways. For the lipid bilayer, the cell membrane consists of a lipid bilayer composed primarily of phospholipids. Phospholipids have a hydrophilic head and hydrophobic tails. The hydrophobic tails face inward, forming a nonpolar region that restricts the movement of polar or charged molecules. This hydrophobic core acts as a barrier to the diffusion of hydrophilic molecules across the membrane. The fluid mosaic model describes the arrangement of lipid and proteins in the cell membrane. The lipid bilayer has fluidity, allowing the lateral movements of phospholipids within the membrane. This fluidity enables the redistribution of lipids, influencing the overall permeability of the membrane. As a result, the membrane can adjust its composition and properties to accommodate different permeable requirements. For integral proteins, they are embedded within the lipid bilayer of the cell membrane, and they can span the entire thing, creating transmembrane channels or pores that allow specific molecules to pass through. These integral proteins contribute to the selective permeability by facilitating the movement of specific substances across the membrane. Different types of integral proteins, such as ion channels and transporters, have specific binding sites or pores that determine the selectivity of molecules they transport. For transport proteins, which are including carrier proteins and channel proteins, play a crucial role in selective permeability. Carrier proteins bind to specific molecules and undergo conformational changes to transport across the membrane. They exhibit selectivity based on the shape and charge of the transported molecule. Channel proteins form pores or channels in the membrane that allow the passive movement of specific ions or molecules based on their size and charge. And then for the selective ion channels are specialized proteins that facilitate the movement of ions across the cell membrane. These channels exhibit a high selectivity for specific ions based on factors such as size, charge, and the presence of specific binding sites. Ion channels enable the regulated movement of ions, contributing to the electrical properties and signaling capabilities of cells. Collectively, the combination of the lipid bilayer and protein components of the cell membrane, along with their specific structural features, results in the selective permeability of molecules. The hydrophobic core of the lipid bilayer restricts the movement of polar or charged molecules, while integral proteins and transport proteins selectively facilitate the transport of specific molecules or ions across the membrane. This selective permeability is vital for maintaining the integral environment of the cell and controlling the movement of substances in and out of the cell. So still we have that cell membrane on the image. So next we have our vocab words. We are defining a lot of words, which include plasma membrane, phospholipid bilayer, extracellular fluid, channel protein, and you get the idea. They're all imaged on the screen. So the vocab words related to the structure and function of the cell membrane include plasma membrane, the outermost boundary of a cell composed of a phospholipid bilayer embedded with proteins, separated the cell from its external environment. The phospholipid bilayer is the main component of the cell membrane, consisting of two layers of phospholipid molecules arranged with their hydrophilic heads facing outward and hydrophobic tails facing inward. The extracellular fluid is the fluid outside the cell, which include interstitial fluid and other bodily fluids. The channel proteins are a type of integral membrane proteins that form channels or pores in the cell membrane, allowing the passage of specific ions or molecules. The semi-permeable refers to the property of the cell membrane, allowing certain substances to pass through while restricting others based on their size, charge, and polarity. Passive transport is the movement of substances across the cell membrane without the input of energy. And then diffusion is the passive movement of molecules or ions from an area of higher concentration to an area of lower concentration, resulting in their even distribution. Concentration gradient is the gradual change in the concentration of a substance from one area to another, driving the movement of molecules or ions during diffusion. Solute is a substance that is dissolved in another substance, which could be a solvent, to form a solution. And then a solvent is a substance in which solutes dissolve to form a solution. That's funny. Simple diffusion is the passive movement of molecules or ions directly through the lipid bilayer of the cell membrane. Facilitated diffusion is the passive movement of molecules or ions across the cell membrane with the assistance of transport proteins, such as channel proteins or carrier proteins. Channel-mediated diffusion is the movement of molecules or ions across the cell membrane through protein channels, which selectively facilitates their transport. Gated transport are protein channels that can be opened or closed in response to specific signals or stimuli, regulating the movement of ions or molecules. Leak channels are channels that are always open or have a baseline level of permeability, allowing the passive movement of specific ions or molecules across the membrane. Carrier-mediated diffusion is the movement of molecules or ions across the cell membrane with the help of carrier proteins, which binds to and transports specific substances. Active transport is the movement of substances across the cell membrane against their concentration gradient, requiring the input of energy, usually in the form of ATP. Pumps are membrane proteins that actively transport ions or molecules across the membrane against their concentration gradient, utilizing energy, usually in the form of ATP, to maintain concentration gradients and perform specific functions. Understanding these vocabulary words will help you grasp the concept related to the structure and functions of the cell membrane. As these words are seen on the screen, those are all of them. And then for my last slide, obviously, I just wanted to show my works cited based on where I got all of my information from for this presentation. I just wanted to say thank you for listening, and I hope you learned something. I hope we can all benefit from this study guide. Thank you so much.